This presentation concerns the paper A Pilot mHealth project for monitoring vital body signals and skin conditions. It is well acknowledged that early detection of abnormalities in the health of individuals using existing technological tools will have direct economic and social benefits. Wearable devices are already used for monitoring vital signals and communicating this information to the user or a database. Nevertheless, issues of price, accuracy, and availability are still of concern. On the other hand, mobile devices like smartphones and tablets, which are used extensively by a rapidly increasing number of users over the world, have sensing elements like microphones and cameras, which can be effectively used to monitor vital signs of the human body. Our intent is to develop a user-friendly, accurate, and easy-to-access system in order to collect and analyze such vital signals, having as a central component the smartphone. The collected information should be centrally stored in a cloud and become available for analysis using machine learning and artificial intelligence. In addition, post-processing and visualization should be available through a user-friendly mobile app. Our project includes the following activities: measurement of heartbeat, body temperature, and oxygen concentration levels in arterial blood using a wireless peripheral mini device, heartbeat detection using a smartphone built-in camera, and finally, early detection of serious skin disorders based on machine learning algorithms. Our detection technique is photoplethysmography, PPG, which uses light to assess both the pulse rate and oxygen concentration of blood in a non-invasive way. The key idea is that as a beam of light passes through a blood vessel, its luminosity is decreased because of absorption and scattering. Due to the variation of the blood volume flowing through the vessel, the intensity of light passing through is changing with time in synchronicity with the blood flow. The increase in blood volume during systole results in a decrease in light intensity, while the inverse happens during diastole. Hence, by monitoring the temporal change in the intensity of the beam of light traversing a thin part of the body, e.g. a fingertip or earlobe, one can measure the pulse rate. PPG can be also used in the reflection mode, as we do in our project, where the backscatter light intensity carries the information concerning the blood flow. The fact that blood which is rich in oxygen absorbs light at a significantly different level as compared to blood with low oxygen concentration allows the extraction of the oxygen concentration of blood from the intensity variation of the PPG signal. As we see in the diagram, the absorption of oxygenated blood near the red part of the visible spectrum is significantly lower than that of the deoxygenated blood. The reverse is true in the near-infrared part of the spectrum. Hence, the measurement at two different wavelengths for which the absorption coefficient is reversed allows the estimation of the oxygen concentration, known as SpO2. It is known that the PPG signal is comprised of an AC component, which fluctuates according to the blood flow variation due to the heart pulsation, and a DC component which mainly depends on the absorption of tissue and bones introduced in the path of light. The ratio R of the AC to DC components of the PPG signal at one wavelength to that at the second wavelength is shown to be correlated to the oxygen concentration in blood SpO2. In this slide, we have the hardware design of our circuit. The part on the left-hand side corresponds to the sensor comprised of the red LED, the IR LED, as well as the phototransistor. The non-contact temperature sensor is shown connected to the circuit on the top left-hand side. Two stages of active filtering and two stages of amplification were implemented in our final design using 4 concatenated operational amplifiers shown in the middle of the circuit. The choice of resistors and capacitors is done based on circuit theory and laboratory experimentation in order to optimize the response received. The ESP microcontroller equipped with Bluetooth and Wi-Fi communication channels is shown on the right-hand side. The circuit includes an ESP microcontroller equipped with Bluetooth and Wi-Fi communication channels, 2 stages of filtering amplification, an infrared LED and a red LED, a phototransistor in the range of red and infrared light spectrum, an SD card for saving data on board in case of connection problems, a 5-volt stabilizer for powering the system, a 3.3-volt voltage regulator for the negative power of the active filters, and indicators for visual and auditory debugging. The overall circuit implementation is depicted in this slide. As shown, this is a breadboard implementation illustrating the interconnections between circuit components and ESP microcontroller. Once the testing procedure is completed, we are planning to print the circuit onto a PCB. The objective of this task is to reduce the size of the circuit, thereby reducing the cost of implementation as well. Here we show a flowchart of all actions taken during the measurement and calculation of either pulse rate or blood oxygenation by the ESP microcontroller. Eventually, once these steps are completed, the final calculations of these 2 quantities are stored in a database given the current timestamp. Here we present typical PPG signals for a healthy young person. The upper one was captured using the IR LED, and one can observe both the AC signal as well as the DC signal level. The lower one corresponds to the measurements taken with the red LED. The systolic and diastolic phases are evident in both pictures. The signals shown correspond to process data which have been smoothed by software written on Python. After the microcontroller sends the data sequence in a CSV text file, the software checks their validity and stores them in memory. Subsequently, with users' commands, the code implements smoothing and then plots the data. Data smoothing is based on the Savitsky-Golay filter library. This filter implementation is based on a moving average and linear regression concepts. The heartbeat rate can be then calculated by correctly identifying the extrema of the PPG signal in a given time window, using a 3-point algorithm. In the specific example shown, the pulse rate is 72 pulses per minute, and the oxygen concentration in blood SpO2 was estimated to be 98%. The temperature measured at the fingertip was 35 degrees Celsius. The accuracy of the prototype circuit was assessed by testing several users under resting and exercising conditions. These measurements were compared against the measurements taken with the manual method, where the index and middle fingers of the right hand were pressed on the opposite wrist and the number of beats within 60 seconds were recorded manually. 6 healthy individuals were tested by recording their pulse rate when at rest and after performing minor exercise, 10 or 20 push-ups. The recorded measurement was compared against the benchmark value, the manual measurement. As seen, the 2 sets of data are in very close agreement. The maximum deviation was 3.6%, which is deemed to be satisfactory for this initial phase of the project. Another approach in measuring the pulse rate and the oxygen concentration is to use the smartphone camera to record a video of the fingertip which is directly attached to the lens. This would be an extremely useful approach when the user does not have access to the wearable peripheral device introduced earlier. During the procedure, the flashlight of the camera is always on during the recording time. The captured video files, MP4 or AVI, from the camera are at this stage of the project transferred to a laptop for video processing and analysis. In the future, this information will be stored in the cloud. Our initial experimental data consists of approximately 40 seconds long videos of the index finger while in contact with the rear camera of the smartphone, having the flashlight on. As light from the LED travels through tissue, it suffers absorption and scattering, and its intensity is modulated by the pulsating flow of blood according to the PPG principle. The reflected light is then recorded by the camera at 30 frames per second, and the video is stored in the phone. The recording is then reduced into its frames, and each frame is subsequently analyzed into its three spectral components: red, green, and blue. For each component, we calculate the mean value of brightness per frame, and then we apply a moving average filter in order to smooth the measurement data. The next step is to identify the peaks in the signal and count their number in a given window. The peaks are identified by calculating the gradient at nearby points to the left and the right of the current evaluation point. This process takes place on data produced by the moving average filter, thus the data are smooth and free of high frequency noise. A particular data point is identified as a peak if the gradient is positive at 3 consecutive points before the peak and negative at 3 consecutive points following the peak. In our code, we experimented with a moving average based on the past 7 samples. Knowing the number of peaks and the time span, the pulse rate can be easily calculated, as in our case where its value is 69 pulses per minute. The method was tested against healthy people who belong to a wide range of age, 17 to 52 years old. The recordings were performed under ambient light in the laboratory. The results are compared against the pulse rate measured using the traditional benchmark approach described earlier. 2 types of smartphones were used, one based on iOS and another one based on Android. It was observed that the deviation is relatively high in some cases, reaching a value of up to 11.7%. Note that the blue component was excluded from the measurements because of its low correlation with pulse rate calculation. Our observation is that the results are susceptible to slight movements of the finger, ambient light conditions, or even the charging level of the smartphone's battery. Consequently, it is highly important that in the near future, all these effects be carefully filtered out from the individual RGB signals, providing thus the ground for more accurate pulse rate calculation. The third task is to detect at an early stage potentially threatening skin conditions, applying machine learning algorithms to images captured with the smartphone. We strongly believe that such an automated diagnosis system could be complementary to the work of experienced physicians, thereby increasing confidence in the results. The smartphone camera is used as a tool to take a photo of the skin, send it to the cloud, Firebase, then get it processed by a machine learning algorithm, and finally determine whether it needs further attention by medical professionals. In this project, we use Yolo version 4 as the main engine for classification of the image under investigation, based on prior training of the algorithm using a database of images from open repositories. Yolo uses convolution neural networks. The methodology used in implementing this particular algorithm include the following: data collection, data processing which involves sorting, labeling, and dataset augmentation, and finally classification. Some preliminary results. The number of classes considered were 3: angioma cherry, squamous cell carcinoma, and healthy skin. For data augmentation, we used a MATLAB code. Image annotation was implemented with the use of an open source software which has the option of saving data into Yolo format. Initially, only the more pronounced infected areas of the skin were annotated. but at a later stage the annotation involved larger image areas. The increase in the number of images per simulation was achieved by using a MATLAB code. The operations involved flipping an image, rotating an image, or adding to it Gaussian noise. From the results, it is evident that the increase of the size of the training set results in an improvement of the detection success rate. The initial results of this project were encouraging. We are currently working on the miniaturization of the hardware circuit and the design and implementation through 3D printing of an ergonomic housing case at low cost. We are also designing an Android iOS mobile app to handle the communication with the peripheral device and display post-process results obtained from the cloud. Further work is needed to allow accurate calculation of blood oxygenation directly from the video captured by the smartphone camera. A recent collaboration with the local oncology center will provide us additional high-resolution images on skin conditions that will allow for better training and more accurate results based on machine learning algorithms. Thank you for your attention.